Well, thank you all for the invitation and the opportunity to share my work. Hopefully you can see my slides. Okay, and so this is kind of a new project for my group. I'm very much in the kind of obesity and cardiovascular dysfunction, but I was excited to share some findings from a recent publication of ours, where we're actually looking at adipose tissue in obesity. And so I'll be walking us through, get a pointer here, walking us through some of our findings using RNA sequencing to look at different gene expression profiles and different adipose depots or fat depots. And then I'll culminate the talk with some potential putative targets in adipose that maybe we can use to combat obesity. So again, thank you for the invitation, and I will dive right into some of the background. So we're all familiar with at least obesity as, I think, a construct in health and disease. But the simple explanation of it really is going from this lean person here to a growth in adipose tissue, which is what we think of when we think of obesity. And this can be done through a variety of ways: a poor diet or a Western style diet, high in calories, fat, and carbohydrates, but also coupled with a sedentary lifestyle, so less movement, so on and so forth. So poor diet, low movement, sedentary lifestyle, and also maybe an underappreciated genetic influence can all result in an individual going from this body type to this more obese body type. And it's actually a little bit easier to go from this direction in this direction than it is to go in this direction. And this is kind of important, and you're probably all very much aware, obesity is affiliated with a litany of other morbidities and mortality rates. And I would be remiss if I didn't bring up, some scary statistics with cardiovascular disease and obesity because of my, most immediate interest in vascular dysfunction. But what we're seeing now, and this is from an earlier epidemiology study in 2012, but this is still kind of tracking, is that in the next 5 years, we'll see a 20-fold increase in cardiovascular disease. And even with the onslaught of GLP-1 agonists, Ozempic and others, this is still kind of in the fold here. So we're moving in the right direction with combating obesity and helping cardiovascular disease, but we're still in some trouble with in the field here. And so bringing us back to adipose, not all adipose is seen as the same. There are very much different adipose or fat depots in the body. And this circle with VAT in it, VAT, is referencing the visceral adipose tissue. So those adipose depots that are surrounding our viscera are seen as kind of the dysfunctional and harmful fats versus, say, the subcutaneous or SAT, as I'll be referencing it in future slides, is seen as less harmful or maybe even protective in some instances. And so these two, VAT versus SAT, visceral versus subcutaneous, is a massively growing area of research that my group is starting to kind of get into. And this data that I'm sharing with you today is really focused on the adipose, but again, we're forward thinking into how this will impact vascular function. And so right now I'll be sharing adipose. And one more thing I want to point out with this is that even though surprisingly less appreciated, even though it's the largest visceral adipose depot, that abdominal fat that we're all very familiar with, the so-called beer gut perhaps, is really, really strongly associated with cardiovascular disease. And there's a lot of investigation into pericardial, epicardial fat and its role in directly impacting the heart. But my group is focused on this mesenteric fat or this abdominal fat. And we used an animal model in these studies and some RNA-seq. And I do want to warn you for the next slide, there are dissected mice on this next slide. So you'll be seeing a sacrificed mouse with dissection. So if you're not keen on seeing something like that, your warning is now, and I'll be changing the slide right now. Okay, so here's what we did to kind of try and get at visceral fat versus subcutaneous fat and what might be going on in the transition or the occurrence of obesity, right? So we're interested in looking at visceral fat versus subcutaneous fat as we compare from lean and obese mice. And so we used a very preclinical kind of basic science study where we put mice in two different groups randomly, a lean group where they're just maintained on their rodent diet, and then another one that is maintained on a high-fat, high-carb diet. And these mice are actually maintained on this diet for 12 to 14 months, so a little over a year. And so it's kind of a prolonged obesity diet. And so, of course, you can see that the mice gain weight, as you would expect. And then we isolated these different adipose depots, subcutaneous and visceral. And this is what this looks like in the mouse. And so to point out exactly where we're getting these adipose depots from, this is the inguinal fat pad or adipose tissue. And when you isolate it, it kind of looks like this. And then for the visceral fat, we use mesenteric fat, or MVAT will reference it. It's taken from that fat that surrounds the intestines and the gut, the GI tract. And so we take it out and it kind of looks like this cauliflower looking shape here. And when you compare these adipose depots from lean and obese mice over that time course, you do have clearly increases in mass in both of those depots. The question becomes then, what is happening in this visceral fat that is maybe not occurring in this subcutaneous fat at the molecular and gene level? And so we ran all these RNA sequencing analysis in collaboration with the cores here at UD, both the DNA and Genotyping Center and the Bioinformatics cores. So Brewster Kingham and Sean Polson were very much involved here at UD. And we did gene expression, a whole bunch of different pathway analysis and network analyses. And then we really wanted to focus on differentially expressed genes. But the first thing that we noted and pointed out was that in this Venn diagram of all the genes that we found to be expressed in these different adipose tissues of lean and obese mice, that there are actually unique genes in each of the four groups. That would be denoted by these little portions of each Venn diagram circle here, or oval, that has these genes all by themselves. And if you can point your attention to the visceral fat from obese mice, there's 814 unique genes just in visceral fat from these obese mice, compared to 600, 300, and less than 200 in the other groups. So that right away is telling us that this fat is certainly influenced. A whole brand new set of genes become expressed in this visceral fat depot when mice are made obese. So we immediately were surprised by this, and we're thinking that, well, we can't fish through like 800 some genes and find out, major targets. That's a massive undertaking to kind of see what's going on here. So we really then wanted to say, okay, well, if there are unique genes, we know obese fat is being affected here. Are there similar genes, however, between these two that are going in equal or opposite directions? Sorry. So in other words, if there's a gene that's expressed in sub-Q that's going up in response to obesity, is that same gene perhaps going down in visceral adipose tissue? This is really where the novelty of this study comes into play, and I'll share with you a little bit more about what we did there. So when we have really stringent statistics, we have two more Venn diagram plots. We have what we're finding is the genes that were downregulated in SAT and upregulated in VAT, and vice versa. The point of this is saying that, okay, if we know that SAT is maybe somewhat protective or adaptive to obesity, and VAT, visceral, becomes dysfunctional. Is there a common gene that's going in different directions that might be a more suitable target than, say, hunting through 814 genes that might be uniquely expressed and finding a target there? Is there common genes in these things that are going in opposite directions? To our surprise, when we did these analyses, we only found 4 genes that fit that kind of profile. In other words, going down in SAT, up in VAT, and vice versa, up in SAT and down in VAT. But even when we made the statistics a little less stringent to see if we could increase the field of players, we only altered by a few in the up in SAT, down in VAT, to I think this is a total of 12 genes maybe. So these 4 genes on the left here are understudied in VAT, or adipose tissue in general. So we don't have an idea. We know that chitin is a calcium gene. This is a heat shock protein, so maybe involved in protein folding and stress responses. This ELO-V6 is a fatty acid synthesis gene, and this is an ion channel, so I'm really interested in that because I'm an electrophysiologist by training. But these 4 genes we're considering as putative targets to pursue further. In other words, can we influence the expression of this gene, say, and knock it down in VAT? Does that help restore adipose function and or vascular function in the fat? And vice versa. If we upregulate these different genes here, in VAT that are shown to be upregulated in SAT, does that help us to restore adipose function, which is more inflamed and visceral? It's more lipolytic, and so that's where the damage is kind of coming in from. Can we reverse some of those known observations, inflammation, lipolysis, by manipulating these genes? And so that was the crux of the study. This is really where the novelty came in, this opposite direction type idea. And so we published this, as I said, in Physiological Genomics. It's in the APS journal, and it's gotten a lot of buzz recently. So it was an APS Select manuscript in December of last year. It also received a press release that same month titled Gene Expression Differences in Fat Tissue May Inform Target Treatment for Obesity. We also got a nice Daily article by Amy Cherry. So we're getting some university recognition for this work. And this is Sean Polson and Mark Shaw, the two that Sean Polson's the director of the bioinformatics and computational core. Mark Shaw works with Brewster Kingham in the genotyping core. We most recently received an editorial focus for this work, also in Physiological Genomics. And so we're pretty excited about this work. It's one of those studies that I wasn't expecting all this attention from, and now an invited Gin Talk, so thank you again. But it's one of those learning things where submit the work and see what buzz it can create. And so I know I'm a long talker and I probably went over the time that you prefer, but I'd like to thank you all again. And I'd like to point out some names. Malak Al-Radi is a PhD student in the lab who's the first author on the work. And again, the Sequencing Center and Bioinformatics Center folks. Also, my funding sources, I'm a project lead in my 5th year on Dave Edwards' cardiovascular COBRE, and some core access awards also from these two centers help support the work. So thank you. And I can keep sharing my screen to answer questions, or I can stop sharing. That's totally up to you, if we have time, of course.